So, really great pleasure to welcome and introduce Pietro Fratta for the next talk. So, Pietro is a professor of cellular and molecular neuroscience at UCL, and he also has a footprint at the Francis Crick Institute, and he comes to us all the way from London, the UK. Pietro has really been a pioneer in the investigation of RNA metabolism alterations in ALS, and specifically those associated with TDP-43. Some of his seminal work includes the identification of the connection between TDP-43 function and UNC13A cryptic splicing. And I really think his work has helped sort of shift the focus of the field on TDP-43 loss of function in addition to the potential gain of function effects. So, Pietro, welcome, and thanks for joining us.
Yeah, great. Thank you very much, Evander, for the introduction and for the invitation to come here. It's really good to come to this community. We're trying to have similar symposia, actually, in UCL. It's not as old as here, so it's good to be learning also how these one-day things can really work well. So I'm really fortunate, actually, for the introduction that people have done, but basically so we try and not get lost in these 30 minutes. I'm going to try and divide the talk kind of leading on to what Lindsay was actually saying about how TDP splicing can actually relate to new proteins and biomarkers and the immune response in ALS. And then I'll talk about therapeutic targets, specific ones, and more general approaches. So this was said already, but just to reinforce the point that we know, and this was really a seminal moment nearly 20 years ago when TDP was identified as the protein that aggregates in ALS. And differently from the nucleins and the other neurodegenerative proteins, it's not just its aggregation, it's the fact that it's usually in the nucleus in these healthy cells, still in patients, if I can find, yeah. But in pathology, it gets depleted, literally sucked out. And this really happens, it's interesting to us in ALS because it happens in all cases other than specific genetic subtypes, SOD1 and FUS. But it goes beyond ALS. Of course, FTD, half of the patients, that was an original finding from Virginia Lee already, have this. But in Alzheimer's, it's becoming increasingly, recognized. And there's other diseases outside the CNS, but still that us as neurologists follow, like IBM, where it's actually as mislocalized as in ALS. Basically, virtually all IBM cases have TDP mislocalization. And what does it do? Well, we've heard a lot from Lindsay. So it binds G, repeats, so we can skip that. But Lindsay was saying how you do a normal clip and you actually get the fact that most of the bindings sit in introns, and this is green TDP on introns. And typically no action occurs there, nothing happens. There's no alternative splicing ongoing. Sometimes there is, but mostly there isn't. But what Phil Wong found, in around nearly 10 years ago, in 2015, is that when you take away TDP, TDP loses its repressive ability, and so there's some sequences in those introns that get included in RNA, and we refer to these as cryptic exons, and they're typically not conserved, right? So you have on the left here the conservation of normal exons, and the cryptic exons are really not conserved. And so because they're not conserved, they often cause frame shift, or they have stop codons, and they end up being degraded. And they're really specific. This is one of our favorite ones, in UNC13A, but many look this way. So on top you have a nice iPSC, normal splicing in iPSC. You get always this junction, and you take away TDP, and this new exon appears, right? And this is so nice in a cell in which you knock down TDP, but it's remarkably similar when you go and look at sequencing from patients, right? So when you sequence just these cells that lack TDP in patients compared to the ones that still have it, you get, an RNA-seq trace, which is pretty similar. So the appearance of a lot of cryptic in the disease cells, and basically none happening in the cells just next by in that same part of tissue that still have TDP. And so the specificity goes even beyond when you take these big resources, which even you're using on, lots of RNA sequencing on brains from multiple centers. This is work early on we did in 2020 with Len Petroselli. It was really surprising how you could actually, even in this low quality, because of brain, post-mortem RNA-seq, you could really specifically detect the cryptics just in the places where you expect it. So just in ALS patients or FTD TDP patients, and just in the affected regions, and for example, not in the cerebellum. And so this is what really leads to our interest. I mean, TDP causes many things, and there's alternative splicing changes that happen that most likely are really important to disease. But the appeal we have to the cryptic exons is the fact that they just appear in those cells, right? And so this has crucial potential implications for biomarkers, right? Because if they are detectable, right, if there's enough of them, they can be really good biomarkers. And also therapeutically, right? It's much easier, if they are relevant, to target something that just appears, in a cell that is diseased, compared to going and reestablish an equilibrium of an alternative splicing that always happens in normal cells. And so, if they play a role in disease, and if there's enough of them, these two things should actually hopefully come to fruition. So do we need biomarkers? Well, we do. We also need diagnostic. We never talk about them. In ALS, maybe that's not that useful. But in FTD, an FTD patient still comes to clinic, and you don't know if they have TDP pathology or not. And similarly in Alzheimer's disease. So of course, when we'll have potential TDP treatments, it'll be important. But even more, it's important to know how much TDP pathology is ongoing, because if we're able to do that well, that could really help clinical trials. And so one of the things, and this is published, I'll go quickly through it, but is that, yes, typically there are cryptic exons and they're out of frame and they contain stop codons, but sometimes, by luck, they're multiples of 3 and they don't contain stop codons. And so are they translated? And so we did this kind of proteogenomic effort, very typical and routine actually in cancer, by which we basically predicted ones that didn't have stop codons, and we predicted what their protein sequence would be, what the trypsin digest would do, and so could we detect them in mass spectrometry? And the quick answer is yes. We detected some, right, in 12 of them. There's definitely probably many more out there, but the sensitivity of our study was able to detect these. And most of these are, in frame, as expected. Some of them are exon skippings in frame. And KCNQ2, I won't touch on this anymore in the talk, but is definitely a heavily studied gene and protein and cryptic event here in Northwestern in Evangelos' lab. And so this was quite exciting. We could detect these, and this is how it looks. One of them, HDFL2 on the Western. You can see, when TDP is there, you just get one band. When you start losing TDP, you start getting this extra band, which is what we refer to the cryptic peptide band. It's not a perfect name because it's a protein with an extra cryptic peptide. It's not just a peptide. And so that's interesting. And Len Petroselli went on, along with Phil Wong, to develop specific antibodies to this cryptic peptide. And when you do this experiment in brain, this is work from Len's lab, you really see that you can divide really well, for example, Alzheimer's with TDP pathology versus not. When you do this in CSF, and this is work from Phil Wong's lab, it's really encouraging data. You can detect it and you can discriminate the categories. I think we're not there yet. This is an incredible first proof of principle. I think the assay will need to get refined, but I'm sure it will, to become even more sensitive. But I think there's hope in using this as a biomarker. But what we really, and so this is the extra thing. So you typically, other than leading to loss of protein, by exon inclusion or skipping, you get novel proteins. And so the next question was, well, if you have these novel peptides, like in cancer, can these actually generate an immune response? And so we teamed up actually with Jenny Jiang at UPenn, and she's developed this really cool method by which she's able to basically assemble MHC type 1 tetramers and load peptides into them and barcode them by DNA. So you have these kind of new tetramers presenting a whole bunch of, you can do hundreds of them, peptides, and you can then see if cells from patients recognize them, and you can fact sort them. And so that's what we did with Jenny, and we did that actually not just with ALS, but with IBM, right? And so you can see here on the x-axis, these are FACS traces, are basically the tetramers. So on the y-axis is a viral tetramer, so kind of a positive control. And you can see that you get cells that recognize them in ALS and IBM much more than in controls, and as quantified here. And it's It's interesting because, of course, you have naive cells recognizing anything in our body. And indeed, it turns out that in controls, typically these are naive cells. They're not expanded, whilst in ALS and IBM, not just there's more of them, but they are actually clonally expanded. So they've seen it and they've responded to it. And this is also how it looks like. This is an example for one in HDGFL2. So you take all the cells, with that subtype, and you actually see that they're pretty specific. They just respond to a peptide in HDGFL2. It's not that these are sticky cells. They don't respond to all the other tetramers with all the other cryptic peptides presented. And so that was really intriguing. And so we, especially in IBM, that's where we saw the HDGFL2 response. So we went back, we used the antibody from Len Petrucelli, and we started asking, do actually IBM patients express cryptic peptides? And indeed they do. So this is a control, and you see there's not much phospho-TDP, there's not much P62 or HDGFL2 cryptic peptide, whilst instead in IBM, you can see actually in the same fiber, you get the co-occurrence of altered TDP, altered P62, which are classical hallmarks of disease, and the cryptic peptide. And what we started actually seeing in pathology is that actually this correlated with the immune infiltrate. And we did proteomics on these patient samples. And indeed, the ones on the right, these are all a series of, let's say, important immune genes. People that had high presence of cryptic peptide had much higher expression of these. But this is just looking at HDGFL2, it's one peptide. And although that's the one that we found for IBM, for ALS we also found a peptide in IGLON5. We don't believe it's this one, and we think it's a burden. And so we did actually RNA-seq on these patient biopsies, and we found that on the left you have controls, and you're seeing in the different colors the cryptic exons. And there's not much happening in controls. The burden is much higher in disease. And indeed, when you take the whole burden of cryptic splicing and you compare it to both antigen presentation and immune infiltrate, so on the top is antigen presentation, on the bottom infiltrate, there's a good correlation. So it really tells us, it doesn't tell us about causality, but it really kind of starts linking these two things, right? So we have cells that respond to these, and it seems that when you have more of this, more immune response is going on. And so I guess the conclusion for this part is that beyond having new peptides, these can have value in biomarkers, and they can be linked definitely to T cells activated to this. So what role this plays in disease? I think this is a big question, and David was kind of putting it out there as well, still to be addressed, but definitely something that should not be ignored. So, and to conclude this, of course, I think it's not about this one or two that we found, it's about more. And so there's a lot of effort in the lab to try and actually extend the palette of cryptic peptides being produced. And so actually to do that, we've done work in not just focusing on cryptic splicing, but focusing on alternative polyadenylation, actually cryptic polyadenylation, meaning we've looked for polyadenylation sites that basically do not exist normally, so are on the left, on the x-axis here. So normally they're not there, but when you knock down TDP, they start appearing. And I'll really go quickly because of time, but we found lots of these, and they come in 3 different flavors. Sometimes like Stathmin-2, so these were typically known. You have a splice junction and a new UTR. Sometimes the reads go in the intron and then they polyadenylate. And sometimes they're just UTR extensions. And these events they happen in patients, and the first 2 categories are really crucial because they're in the coding region, so they're really predicted to be stable and to generate new peptides. So we're really interested in these because it may allow us to test much more broadly what immunogenic things could be there in patients. And instead, in these extension ones, I'll just tell you and skip down the slide because of time, but basically what we found is that having longer UTRs actually leads to more RNA and more translation, right? And so basically, in addition to getting cryptic splicing to reduce protein, cryptic splicing to generate new protein, there's the fact that we can actually get cryptic polyadenylation, and this can actually lead to increase of normal protein. And it was our paper that came out coordinated with other 2 groups in bioRxiv, and actually these findings align really well with work from both Evangelos and Sami Barmada. So there's a lot of focus right now on these UTR changes due to TDP. So that was on the peptide side, and then the fact that, well, is cryptic splicing a good target for ALS therapeutics? And so I'm going to really now focus on the fact that the big question, and it wasn't just was, is there's many of these events, right? Great, but do they act as in a concerted manner? Is it the sum of them, or actually can single one of these be relevant? And early on, we focused in one of these events, which is in UNC13A. On the left, I already showed you, it's a cryptic splicing, which is really specific. And it's of the category of the ones that has a stop codon and leads to loss of protein, right? So here, a Western blot here, you lose TDP, you lose UNC13A, pretty massively. And not just in cells, even in patients. And why that was really an obvious thing to pursue is because UNC13A is the second strongest hit in ALS GWAS studies, and the SNPs, actually two of the five that lead the GWAS signal, are in the intron where the cryptic exon is, and actually one of them is in the cryptic exon itself. And so we basically, our work, which I'm just going to summarize, led to identifying this model, which is TDP normally inhibits this cryptic splicing, and you don't have it. Even if you have the risk SNPs, you don't get it. And if you get ALS, even if you have the protective SNP, you still get the cryptic exon. But what changes is how quickly this kind of comes up. So if you have the risk SNP, you get more cryptic earlier on. And if you have the protective, it's a bit slower. It's not a huge change, but actually this change is linked to the progression of disease. You get faster progression if you have the risk SNP. And so, of course, an obvious question therapeutically is, well, what happens if you just get rid of it all, right? And, I think at least we should bring individuals from the faster to the slower progression. That's kind of the minimal, if it's not toxic and if it does what it should. But then it's the question that I have no answer to is, could it actually do a bit more? And so we've done work in trying to take this forward with ASOs. But before that, it's about what does UNC13A do? It's an uncoordinated, that's the phenotype it has in C. elegans. But actually what it does really is that it's really important for synaptic vesicle release. And so what was known from the literature in mice is that these are, each spike here is the release of a vesicle, right? It's a miniature excitatory postsynaptic. So it's not really like proper transmission, it's spontaneous release of vesicles. And when you knock out in mice UNC13A, you get a loss of it. So we reproduce this in iPSC cells. So again, normally you get these erratic spontaneous releases, which really go down when you knock down UNC13A. And when you knock down TDP, the phenotype is really similar. So that was encouraging, but of course, when you knock down UNC13A, you change hundreds of other cryptics. So is it really related to UNC13A? And so what we did is we basically generated cells in which we deleted genomically the cryptic exon, right? So that's not there anymore. And so when you knock down TDP, you don't get the cryptic exon anymore. Normally you get this high band, that's the cryptic, you don't get it anymore. But you still get all the other hundreds and thousands of splicing changes, right? So you're just changing one thing and you rescue UNC13A protein. And what was quite striking, I must say, was the fact that indeed when you do that, you pretty much rescue this very specific phenotype of synaptic vesicle release. So that was encouraging, but of course, although deleting it genomically could be a therapeutic strategy, but it's a bit kind of further down the line. ASOs are definitely a more established and safer way to go. And so we designed a number of ASOs, and we were able indeed to correct the cryptic splicing. This is the cryptic splicing. When you add the ASOs, you basically, you don't reduce it to zero, but you strongly reduce it, and you rescue the protein levels a lot. And so it was quite nice to see that the treatment with ASOs rescues these nice spikes. So basically, at least in vitro, it shows that the synaptic function can go back to as it was. And so the conclusions here are that's what we're doing. We're working now in collaboration with a company called Trace to actually bring this to patients. And with ASOs, there's the hope that this will happen right in the next 2 years. So that's really exciting to see it progress so quickly. And the last 6-7 minutes I have, I'll actually switch gears slightly and say, fine, it's great to correct UNC13A, it's great to correct STATMIN2, KCNQ2, which is a home favorite here, would be greatly appreciated if that were corrected to in patients, but couldn't we actually go broader, right? And could we actually use the cryptic splicing event to our own advantage? And I think the premise here is the fact that in a dish, we have lots of great things that cure ALS, right? We have a nice diagram of an ALS cell with aggregation and depletion, and these are just 5 great papers that show approaches that can rescue the situation. But the problem is the fact that it's just few neurons that have pathology in patients. Most neurons are still normal, right? So we have billions of cells, and what happens is if we give a gene therapy, even limiting it to neurons, we're going to express it in all these cells. And this is a bit of a problem because although it may rescue the few cells that have disease, and that's great, a long-term expression of pretty much anything can be toxic. We've learned that from SMN, great protein, people lack it, but actually, if you overexpress it for too long, even SMN can be toxic. And so what would be ideal is actually if we could get the beneficial effect by just activating the therapy in cells that have the pathology and have it turn off when the job is done, but actually leaving the other cells, let them be how they are. And that would allow us to be even bolder in what we can give cells, right? Because if it's just limited to those few cells, we can actually think to be a bit more aggressive and think, well, you know, let's hit them hard to rescue them. And so to do that, we thought, well, can we not take advantage of cryptic splicing? So what I've said now a number of times, but I'll show again, is that in the red cells, the path cells, you get the cryptic exons, and typically, like in UNC13A, this leads to a stop codon and loss of protein. And we thought, can we actually do the reverse? Can we do a gene therapy that when TDP is there, actually codes for a nonsense gene therapy? So you get no gene therapy expressed. But when TDP is lost, only then does it include all the exons, and then the gene therapy gets produced. And so we started exemplifying that just by expressing no gene therapies, but red fluorescent proteins, M scarlet or M cherry. And typically we, in biology, right, we deliver these as a cDNA. So you express it and every cell gets red, regardless of whether they have TDP or not. But we thought, well, why don't we stick a cryptic exon in the middle of the sequence? In this case, it's M scarlet. And so that should lead to a normal cell with TDP to assembling an imperfect RNA, no red, but actually if the cell lacks TDP, it gets all 3 pieces and can code for a red protein. And so Oscar Wilkins led this project at the lab to actually devise how to generate exons in whatever sequence you want, which is quite complex. And so he devised this kind of in silico directed evolution using Splice AI to do that. And it works. It works pretty well. So if you express a constitutive red, of course, you get all red cells, whether they have TDP or not, minus and plus. But if you put the cryptic in it, you just get the red when you lose TDP-43. And this doesn't just work in cells, it also works in mice. So when you inject this under an AAV format in mice, normal mice on the left, you get no red. These are the motor neurons in the spinal cord, lots of yellow signal, which is TDP, and no M cherry in this case. But when you inject it in mice that lack TDP just in the motor neurons, you see it starting to become very red, and it activates in the cells where it's supposed to. So that was really encouraging. And but of course, it's not about expressing red, although that can be useful for as a biological tool to study mice. But we thought, what can we actually express in these cells to rescue a phenotype? And the idea was to use something that was actually not developed by us, but by Phil Wong. I think it was a great idea. So he, in a 2015 paper, devised this chimeric protein that has TDP binding. So it binds where TDP needs to RNA, but it doesn't have the C-terminal part of TDP, which is the one that makes it aggregate. So in theory, it's kind of a TDP function-like protein, but it doesn't get sucked in aggregates. And it should be pretty innocuous, but you don't really want to express it a lot. And so we thought, well, why don't we stick a cryptic exon in it to regulate it? And so again, it works, meaning that you get the cryptic exon just when you lose TDP, And so on the Western blot, this is the protein. You just get it when you lose TDP. It's not expressed when TDP is there. Of course, the constitutive one is there all the time. And it does create an advantage. So when you take cells and you express the constitutive or the cryptic version, actually the constitutive ones here in red shows that it has a growth loss. So it means that even if it's not a dramatically toxic protein, if you express it in cells, it does cause some problems. So instead, if you have a regulated version, cells grow exactly like cells which have received a fluorescent protein, in this case BFP. And so we thought, does it have an effect? Well, it does. It can rescue cryptic splicing. You can see here, this is our favorite event, UNC13A, cryptic exon, no normal. If you express both the constitutive or these regulated versions, you don't get it to 0, but you rescue normal cryptic splicing a lot. And but the benefit is that it works beyond UNC13A, right? It should, in theory, rescue everything. Here I'm showing examples of how it rescues well UNC13A and many others. ARS1 is an example, but willingly we put here an example of STATMIN2, another important cryptic event, in which there is rescue of nearly 50%, but by no means it's perfect. It's not normalized. So I think we can do better in what we put there. But yeah, I think it shows well how it's starting to get a broader rescue of these events and not just one. And so that's what we envision, a therapy that you give. You could even give in a C9 person before it develops disease because it stays quiet, it does nothing. But then when some cell starts getting disease, that's when they start expressing it and hopefully correct the deficits soon. And this is just a final summary in which this is how we envision this platform, right? So you have TDP pathology, cytoplasmic gain of function and loss of function. With ASOs, we can correct specific things downstream of this, and I think this will happen in the clinic soon and could be really beneficial, hopefully. But this platform, which I think will take a bit more to get to patients, could actually allow us not just to correct the nuclear loss of function, but express chaperones, express or knock down other factors like we've seen in earlier talks to actually address the gain of function as well. And so with this, I'm finishing on 25 minutes and 50. And on the left, there's all the wonderful people in the lab who've done the work. In orange down are all the UCL collaborators. It's a great neuroscience and ALS community. And on the right, a number of people, both in London, mostly here in the US, some here at Northwestern, with whom we're kind of sharing this exciting journey. So thanks for listening and thanks for having me here.
So the floor is open for questions. Maybe I can take one from the chat. They ask whether those cryptic peptides could be detected in exosomes.
So they could be, and there's a lot of active work in that. We are, unwillingly getting to do some exosome work because I still really haven't understood what they are, but not on the proteomic side. So I don't have any primary data to show that, but it could be.
Dr. Kalb has a question, yeah. Oh, it's not working.
That's amazing. I guess my question is, so when you get a very substantial reduction in nuclear TDP-43, you get these splicing events. I'm wondering whether the threshold for engagement in your transgene is going to match the reduction in which will actually occur in patients.
Yeah, well, I didn't get to, and we're definitely conscious, and a lot of the lab is all working on thresholds, actually. So you can tune it, you can do it how you want. So, that's actually a nice thing, it's modulable. So you think, well, if you're expressing a chaperone or an innocuous thing, relatively, you can be a bit generous and say, well, let's activate it really early on. If you wanna express a genome editor, maybe you want to be much safer and lower the threshold, meaning you just express it really when a cell is in distress. So in that sense, I don't know if that addresses your question, but but there's always going to be a trade-off, of course. If you are sensitive, there's going to be some leakiness. And I think there's no way out of that. An appealing thing of Phil Wong's or similar approaches of chimeric proteins is that they can auto-regulate themselves. So you can afford to be a bit sensitive, and then what you're expressing is anyways gonna limit its own expression. And so in that case, you're a bit safe. You're not gonna overburden the cell with something. But that's not valid for everything. Yeah.
Can you hear me?
I can repeat the question.
When do you think in disease these cryptic peptides show up? Do you think there's any chance they could be like in pre-symptomatic individuals?
So Phil Wong showed that they are. So in his work, he shows using C9 people actually. He concludes that there are even higher in pre-symptomatic. I think maybe that's still to be defined. But you know, the life cycle of a cell is different, of course, from the one of a patient. So when a patient comes to clinic, and you know, we say that a significant part, I won't put a number of motor neurons, have already died. Well, they've gone through a whole cycle of total loss of TDP. So that person has clearly already had cryptic exons and peptides.
Cool.
Although,
thanks.
yeah.
Really fascinating, Pietro. I was really intrigued by the immunoreactivity of these peptides, so I just had a couple of questions about that. So I imagine you've tested a number of these. Do you see that only some are immunoreactive? And are you assessing the specific cryptic exon that's included, or are you assessing the entire protein? How are you doing these experiments?
So basically, we've assessed until now experimentally 18 of them. I'm happy to show the list, and they're not all positive, meaning some of them were chosen, like Stathmin-2, as probably kind of negative, and they showed up to be negative. Then what we do is, that's our part, and we define the peptide sequence, and then what Sean and Jenny's lab is, they use a number of pretty established in the field ways to actually predict which ones should be the best epitopes out of that sequence. And all of those we try and load in the tetramer assay. And so what we got is, so I say HGFL2 and Niglon5 because those are the ones that we identified, clonally expanded. We recloned in other T cells and showed that they activate and they do the whole thing. But we got a reaction for many more than 2, for at least nearly half of the 18 we tested. But you know, I think it's early days to conclude about those. I think we need to test more and validate more.
I see. And do you think this happens because these are de novo events rather than sort of isoform switching events?
Well, so coming to this, in my total ignorance of immunology, that was my thoughts, and I thought, of course, they have to be de novo, otherwise there's going to be some. But you know, speaking to people in the field, you actually, you know, I was speaking actually to Larry Stern at UMass. He was really excited about actually the upregulation in polyadenylation because, you know, in his view, I'm kind of reporting, even massive upregulation of wild-type peptides can actually bring them beyond the threshold and induce immune responses. So I think we have to be, I definitely had to evolve my knowledge of medical school immunology in the last year, yeah. Things have changed.
Maybe one more question.
Yeah.
So these cryptic proteins, do they have any role in synaptic function or synaptic dysregulation, like spine morphology, disintegration, or any neuronal connectivity? That was the other question.
The proteins itself, we detected in the peptides some postsynaptic proteins in Caldron. Not synaptic on excitability, that's the work Evangelos is doing, and that's another, call it peptide, that's a different, it's a new kind of epitope. But they're not more enriched than the splicing in that category.
Thank you so much.
Thank you.
Thank you. So this brings us to the end of the morning session, and I'll try to do my best to put things together and in perspective. So as you can see, we try to focus on TDP-43. That's a key protein, and We discussed its role in the nucleus and in the cytoplasm. And Dr. Kalb investigated TDP's effect in UPS, and he actually found two proteins that when their expression is reduced may actually have a potential solution for the TDP, because he concentrated on the UPS, which is ubiquitinated protein system and protein aggregation. And Dr. Lindsay Hayes actually studied the nucleus cytoplasm transport of TDP and how that is affected in disease. And she identified the cryptic proteins, and that those cryptic proteins were under investigation. And then Dr. Fratta actually showed us data to show that these cryptic proteins play a role in neuroimmune modulation, and that they can be further utilized as a biomarker, and that they also help us understand the disease pathology. And then there are ways to correct them with ASOs. And this also ties, especially the neuroimmune part, to Dr. Gates, who actually showed very clear evidence of neuroimmune component in ALS using samples actually obtained not from mice or other disease models, but from human. And that's very disease related. And Dr. Marco Martina showed us how cortex is complex in ALS, and that the interneurons also play a role in the function of the upper motor neurons, the interneurons also affecting those. And then I presented the role of TDP pathology in mitochondria and ER, that when we have TDP pathology, we also have mitochondrial problems and ER problems. And together with Dr. Silverman, we actually have a new compound trying to mitigate those problems in an effort to reduce TDP pathology. And as you can see, big problems require collaborations and team effort. And today here, we try to bring different minds and different expertise all together, because ALS is a complex disease and we have to attack ALS from many different angles. And that's why we are here today, and our hope is that after this symposium, you talk to each other, you start new collaborations, and we want to bring the team together so that we can actually win this battle against ALS.